in metabolic engineering, we're going to pick up where we left off before on metabolic flux analysis, specifically C13 flux analysis. And then we're going to make our way into sort of this next unit that's really focused on gene regulation, protein expression, and using naturally evolved regulatory machinery to be able to control metabolic pathways, not just from a simple overproduction standpoint, but even getting into sensing your product, creating innovative design strategies to maybe use logic or feedback to regulate your different pathways. So that's where we're headed. I go back to about where we left off. Oh, actually, I'm just doing that on my screen. I need to share my screen. Let's see, share screen. This. All right, cool. I will take it that you guys can all see that. And I'll go full screen mode. Okay. So in C13 flux analysis, we've kind of assumed with the title that what we're talking about is a labeled carbon isotope. But in part of today's lecture, we'll talk about how this strategy can be applied a little bit more broadly. I mean, in general, it works for any small range of isotopes that fit a certain set of assumptions. Primarily that they don't disturb metabolism as it ordinarily is. Otherwise, the introduction of your tracer could bias your results. And so in terms of thinking about how it might affect metabolism, but also just in general, how this kind of analysis is done, you have your isotopes and you supply them to your cells or whatever your organism may be. And then you use an analytical technique. And you can use any one of 13C-NMR, GCMS, LCMS, looking at different kinds of outputs, obviously. And for those of you who have had analytical chemistry or maybe even organic chemistry, you'd be familiar with these kind of patterns. This, for example, is a fragmentation pattern of mass over charge. And so if you have, for example, effectively an extra neutron, you have a higher mass, the same charge. And so that's kind of what you're monitoring here. Now, key to thinking about, in particular, 13C and isotope oriented flux analysis is that these are refinement techniques, right? So you're not trying to map out all the fluxes of a new organism from scratch. You have some flux measurements that you've already done, because in some of your pathways, for example, you might not have needed an isotope. If you recall, the underdetermined nature of your fluxes comes from, especially when you have multiple routes, say in this case to GAP, you could go through glycolysis, you could go off through the pentose phosphate pathway and come back around, and you don't necessarily know which, just from GAP levels alone, you may not be able to track which route, what level of flux went through. So when you do your isotope labeling, you get a particular pattern through each route, and then you can effectively apply a least squares regression approach to say, here are my measurements, this is the fluxes that I'm being told, this is what's simulated, and so I see these differences, and so I'm just going to try to minimize the differences or the sum of differences. And so that is the approach that's taken. We covered a slide that was virtually identical to this last time about the history of different isotope tracer studies. And so I won't really dwell on it much here, other than a lot of the developments, particularly in applying it to metabolic engineering, have been in the 2000s, and many pioneered by Professor Antoniewicz. So I've sort of talked generally about what kind of approach one might use, but not about specific compounds. or about the method, for example, of quenching cells and making these measurements. I won't go into all of those details today, and some of them, frankly, are not all familiar to me. But in regard to tracers, it's good to know that you have a variety of options in terms of what metabolite you start with. So you could start with simple sugar, but sometimes that's not relevant for the particular pathways that are underdetermined that you'd like to study. You could start with something like CO2, and that can be tricky because if there are organisms that, for example, use CO2 as their sole carbon source, you've only got one carbon, and if they require it, then you have to come up with an innovative strategy to account for the fact that your only source of carbon is labeled. You know, typically you want still some variety of 12 carbon and 13 carbon isotopes. But in fact, you know, people recently in the field have pioneered more dynamic strategies for dealing with exactly the problem that I just described of the, you know, carbon dioxide utilization. So you also have a choice of what element you want to use as your isotope. This lecture is basically entitled 13C. You also have deuterium, 15N, 18O, and you might find that your prices vary considerably. These kind of price ranges, $100 to $400 per gram. It's also very common of the kinds of molecules that my lab tends to work with a lot, which are non-standard amino acids. They also can vary in this price range. And really what the two have in common that results in this higher price is usually the application of a synthetic method, organic chemical synthesis, to make these compounds. So what's shown here in this box are different forms of pyruvate, where you've essentially got a different degree of 13C labeling. And you can see how that effectively shifts your mass. And so there's just some details here about how these are labeled, where this, in the traditional parlance, there's maybe a 3-digit code for your 3 carbons, and here you can see which of them is labeled with your 13C, effectively a binary code. And then you've got the number of labeled atoms showing up here. Okay, so you supply some molecule, it could be pyruvate, or in this case, a 4-carbon molecule, and your cells do some metabolism with it. And then buried into this step is you've really, for almost any kind of metabolic engineering analysis, where you want to track fluxes carefully and really monitor what some people refer to as pulse chase or other kinds of experiments where the amount of time that something is happening over is really critical. You'll want to do some kind of fast quenching method. And there are a number available that use, for example, really cold methanol. So we're talking methanol that maybe has been stored at -20C or even -80C. And so, or your cells may be exposed to that temperature briefly, and then methanol is used in some cases both to lyse the cells or after lysis, in both cases to perform an extraction. And then, depending on what your metabolites are, particularly for GC analysis, you may do some kind of derivatization step to make it go through the column more appropriately. And I think there's a slide on that later. So here's just an illustration of the different kinds of mass isotopomers you could get for 5-carbon molecule. And you know, this is a study, for example, where presumably you're comparing your wild type organism and its flux distribution versus a mutant, maybe a single gene knockout, and you're trying to investigate the role of that on some fluxes. And so you can see, for example, how for different metabolites, you've got some shifting of your ratios here